Hi friends, good morning. How are you? Hope all are fine. Welcome to Sam online class. This is Dr. Subrat Kumar Mandy. Today we shall discuss about Phagemid. Phagemid is a plasmid that contains an origin of replication from a bacteriophage in addition to that of a plasmid. As an example, we will discuss about P blue script 2 SK plus and minus. This is a Phagemid vector which has 2961 base pair, and this Phagemid is derived from plasmid PUC19. So the Phagemid contain two origins of replication. One origin of replication from plasmid, other origin of replication is from a bacteriophage. So what are the functional modules that are present in a Phagemid? First is origin of replication. As you already know, all Phagemids contain two origin of replication. This origin of replication is from a plasmid, and this origin of replication is from a bacteriophage known as F1 phage. In this Phagemid, there is a portion of the lag Z gene. In this Phagemid, there is a portion of the lag Z gene. And within the lag Z gene, there is a MCA sequence. MCA sequence means multiple cloning site sequence or polylinker. This multiple cloning site sequence contains restriction site for several restriction enzymes. So this is a part of the lag Z gene of E. coli. And this part of the lag Z gene, that is known as lag Z alpha, it allows for the alpha complementation of lag Z gene within the E. coli cells in which the lag Z alpha function is lacking. So this Phagemid contains a part of the lag Z gene. This part of the lag Z gene code for the N-terminal segment of the enzyme beta-galactosidase, and the presence of this part of the lag Z gene, it provides for alpha complementation within the E. coli cells in which lag Z alpha function is lacking. Next is this MCA site. This multiple cloning sequence is flanked by two promoters. On this site, there is a phase T3 promoter, and on this site, there is a phase T7 promoters. So there are two promoters, phase promoters, that are flanking the MCA sequence. On this side, there is a T3 promoter that is derived from T3 phase, and on that side, there is another promoter that is derived from T7 phase. Next is this Phagemid also contains a antibiotic resistance gene. This is the antibiotic resistance gene. This antibiotic resistance gene, it provides ampicillin resistance. This gene, this is a antibiotic resistance gene that provides for ampicillin resistance. And this Phagemid is a multi-purpose vector. The Phagemid P blue script 2 SK plus and minus is a multi-purpose vector because it can be used as a cloning vector, It can also be used as expression vector. Similarly, this Phagemid can also be used as a riboprobe vector and also as a sequencing vector. So, Phagemid P blue script 2 Sk plus and minus is a multi-purpose vector. The DNA insert can be integrated in a specific orientation within the MCA sequence by using two different restriction enzymes that generate two cohesive ends. Same two different restriction enzymes are also used to cleave the MCA sequence for integration of the DNA insert. In this Phagemid, the orientation of the MCA sequence is such that transcription of the log Z gene always proceeds from SAC1 restriction site to KPN1 restriction site. Such a Phagemid is called as P blue script Sk plus and minus. The orientation of the MCA sequence can also be reversed. In that case, transcription of the log Z gene will proceed from KPN1 restriction site to SAC1 restriction site. Such type of Phagemid will be called as P blue script Ks plus and minus. If the MCA sequence is oriented in a reverse manner, in that case, the transcription of the log Z gene will proceed from KPN1 restriction site to SAC1 restriction site. Such a Phagemid will be called as P blue script Ks plus and minus. After integrating the DNA insert within the MCA sequence, this recombinant vector is introduced into E. coli cells from which the log Z alpha function is lacking. Inside the E. coli cell, this recombinant vector replicate and propagate like a plasmid. This Phagemid can also be used as an expression vector because the DNA insert which has been integrated within the MCA sequence is also transcribed along with log Z gene that result in the formation of a fusion protein. This fusion protein contains a part of the polypeptide chain that is derived from DNA insert and remaining part is derived from the log Z alpha gene. So in this way, this Phagemid can also be used as an expression vector. Phagemid is also useful as riboprobe vector. In this vector map, you see this MCA sequence is flanked by two promoters. One is T3 promoter, other is T7 promoter. T3 promoter is from T3 phase, T7 promoter is derived from T7 phase. So by using the T3 promoter and T7 promoter, which are flanking the MCA sequence, RNA copies of the either strand of the DNA insert can be generated. These RNA copies can be labeled with radioactivity or it can be labeled with non-radioactive label. Such RNA copies can be used as riboprobe. So this P blue script Sk plus and minus is also used as a riboprobe vector. Phagemid is also useful as a sequencing vector because by using this Phagemid we can generate the single-stranded copy of the DNA insert as well as that of the vector. So to obtain single-stranded copy, the transformed E. coli or the E. coli that contain this recombinant vector has to be infected by F1 helper phage. The origin of replication of the F1 phage allow for rolling circle replication. So by orienting F1 origin of replication in opposite direction, one can generate the single-stranded DNA copy of the either strands. So these single-stranded copies are very useful for DNA sequencing. So friends, this Phagemid is a multi-purpose vector. That means it can be used for cloning. It is also used as expression vector. It is also used as riboprobe vector and also as a sequencing vector. Thank you friends. Please share, like and subscribe this video, and also please give your comments. Thank you.